Sauerkraut is a dish made of fermented cabbage that has been made for centuries. It is most commonly found in Europe, but can be eaten and made all around the world today. Sauerkraut fermentation is traditionally a spontaneous fermentation that relies on lactic acid bacteria.
So let's start by talking about the process of making sauerkraut. It's a pretty simple process. All you're gonna need is a cabbage, some non-iodized salt, a bowl, a cutting board, a knife, an empty and clean mason jar, a weight, and an airlock lid. So first of all, you have to remove the outermost layers of the cabbage and discard the core. Then you can cut and shred the cabbage leaves and massage them in a bowl with the non-iodized salt in order to extract all of the liquid out. The salt must be non-iodized because iodine inhibits lactic acid bacteria growth, which we don't want. Once there's enough liquid extracted, you can transfer both the cabbage and the liquid in a mason jar and place one of the larger outer leaves of the cabbage over the surface of the sliced cabbage. Then you can put a weight on top of it. Both the larger leaf and the weight will help to keep the cabbage submerged in its liquid. Afterwards, you can close the mason jar with an airlock lid. The airlock will prevent pressure buildup due to the production of carbon dioxide during the fermentation. Finally, all you need to do is store your mason jar in a dark place at cool room temperature and wait for about 3 weeks.
The most important and dominant microorganisms in sauerkraut fermentation are lactic acid bacteria, which are naturally present in cabbage. They are the most dominant because of favorable fermentation conditions, which tend to be high salt anaerobic conditions around 18 to 20 degrees Celsius. Lactic acid bacteria also produce antimicrobial metabolites which stop the growth of pathogenic and spoilage microorganisms, specifically fungi and several gram-negative and gram-positive bacteria. These mechanisms help lactic acid bacteria to outgrow the other microorganisms and help preserve the cabbage. The antimicrobials produced by lactic acid bacteria can be divided into 3 categories: bacteriocins, organic acids, and other small molecules. Bacteriocins are antimicrobial peptides that can destroy microbial strains that are similar to the strain it was produced by. They also inhibit the growth of pathogenic bacteria by inhibiting peptidoglycan synthesis, which is a component of a bacterial cell wall. Bacteriocins cannot be detected by smell or taste in food and are safe to eat as they are cleared by the enzymes in our digestive system. The organic acids produced by lactic acid bacteria, such as lactic acid, formic acid, and succinic acid, inhibit the growth of several gram-negative and gram-positive bacteria, as well as yeast and molds. This helps the sauerkraut to avoid contamination from unwanted or wild microorganisms. Other small molecules produced by lactic acid bacteria include diacetyl, hydrogen peroxide, and rutherine. Diacetyl and rutherine alter metabolic processes of bacteria by deactivating key enzymes, while hydrogen peroxide destroys membrane proteins of gram-negative bacteria. Let's talk about lactic acid bacteria metabolism. The primary fermentable sugars in fresh cabbage are glucose and fructose. Sucrose is also present, but at a much smaller amount. Glucose is consumed as an energy source for the lactic acid bacteria and converted into lactic acid, acetic acid, and mannitol. In early fermentation, the dominant lactic acid bacteria are heterofermentative leuconostocs because they are tolerant to low acid conditions. Over time, as pH conditions drop and acid levels rise, the environment is dominated by acid-tolerant lactobacillus. Other dominant species in the fermentation are Clostridium, Enterobacter, and Weissella. These species mainly contribute to the production of key flavor compounds. Those are all the processes and science behind the making of sauerkraut. Thank you for watching and enjoy your sauerkraut.